Hello everybody and welcome to this presentation on dynamic optimization strategies towards addressing the challenges in protein therapeutics. My name is Heuristic. I'm a PhD student at RWTH Aachen University and I'm really excited to introduce you to our work. Protein therapeutics have emerged since the early 1980s and by now they have become a leading sector in the pharmaceutical industry with a wide range of applications. There are quite a few challenges related to their production, among which an increasing market demand, which has led to the past years in a significant increase in product titers. We have many patents expiring, which has led to the advent of biosimilar and bio-better products. And we have regulatory authorities pushing towards the direction of incorporating quality by design principles. All these point to the direction of flexible operation, decreasing cost of goods, and shifting the focus from developing new processes to understanding and optimizing existing ones. And for this, there are mainly two approaches. On the one hand side, we have trial and error methods, which are costly and time consuming. And on the other hand, we have the development of model-based techniques, which upon their successful utilization, they can provide a real time to market advantage. For the scope of this presentation, we're going to focus on dynamic optimization strategies for quantitatively and qualitatively improving the performance of monoclonal antibodies production, since monoclonal antibodies constitute a dominant product within protein therapeutics. And we're going to focus on answering mainly three questions. The first one has to do with modeling and what changes when we create a model to describe process behavior versus using a model to perform computationally intense optimization studies. In the next step, we're going to talk about multimodality and how to find ways to avoid convergence to suboptimal local solutions, which happens due to the highly nonlinear and non-convex behavior of the underlying models. And the third questions we're going to try to answer is what to do when we care about product quality and how to incorporate it into our optimization problem, since when we talk about high-value biopharmaceutical products, quality, of course, matters. But let us start with the current status in modeling of monoclonal antibodies production, and this is typically done in mammalian cell cultures due to their ability to perform post-translational modifications in an effective way. And you can find in the literature different papers providing an overview of those models. Typically, they are divided into They can be categorized based on whether or not we include intracellular compartmentalization, leading to structured models, and whether or not they include cell heterogeneity. If yes, we have segregated model, otherwise we have unsegregated models. And as the level of detail increases in those models, of course, the computational effort we need to simulate and optimize with this model also increases. So this can be also a limiting factor. Recently, also models that describe quality attributes such as glycosylation have been developed in the literature, and those models have been also used for optimization studies in order to improve the process performance. Of course, as most of these are too complex to be solved analytically, computational algorithms are required for this scope. To provide an example of the influence of the modeling structure into an optimization problem, we performed the following example. We took an unstructured, unsegregated, energy-based model based on cell metabolism and energy balances from our collaborators at Imperial College London. And we applied to that model different reformulation and reduction steps consisting of function smoothing, proper scaling, and removing parts that were not used within the optimization. And then we performed optimization with the original and the reformulated model. And here you can see the results of this comparison. We can see that in blue the original model and in red the reformulated one, and we can see how the reformulated model can yield to a better objective value. More importantly, it can terminate with satisfying all quality criteria, which was not the case for the original model. And also this can be done with with the 1/4 of the computational time that was needed for the original model. So by now we've seen that formulation matters when we talk about computationally intensive optimization studies. And in the next step, we're going to move to our second question, which has to do with multimodality. And I'm trying to illustrate this problem here to you and propose a stochastic approach to tackle this problem. So we have our model and we have our objective function, which can be bi-objective formulation. First, we give to this weight parameter alpha the value of 0 so that we only have one objective, which is the same as before, to optimize the product titer. And in the next step, we also add this term, which is the viability term, and has to do with the percentage of viable cell into the culture. And we can see here that as we incorporate viability into the objective function, we can achieve better results in terms of our first objective, which is only possible due to the existence of suboptimal local solution. So here we have an example that multimodality arises, and we have a way by incorporating biological knowledge into the optimization to find superior local solutions. Another way to tackle multimodality is problem decomposition. So we have our optimization problems. Now we want to again maximize the product concentration at the final time. And for that we can vary initial concentrations and feeding profiles. And we split that problem into two optimization problems, one from optimizing initial concentrations and the second one for optimizing the feeding profiles. And here in the intermediate step, we enable of different objectives. And as we do so, we see that if we use the same objective for the intermediate steps, we kind of find a similar solution to what we would find without decomposing the problem, which is actually reasonable. But when we here use a different objective, and in that case we use the number of viable cells into the culture, we saw that we were able to find again a better solution at the final time. This again shows the multimodality of the existing problem, and is only possible because of local solutions, because otherwise we would expect that the first approach that we from the beginning can give as degrees of freedom to optimize everything should yield the best objective, but it doesn't, because we have nonlinear models that can have multiple local solutions. And we see that by decomposing the problem and adding a higher degree of flexibility to the optimization problem, we can again find superior local solutions. Up to now, I've shown you a stochastic and a heuristic approach to tackle multimodality, and although both methods are good in finding, in terms of finding superior local solutions, their disadvantage is that in finite time they cannot guarantee convergence to the global solution. And as this might be wished, here we show an alternative approach which has to do with deterministic global optimization, which is the only way to guarantee that our solution is the best solution we can ever find. However, a deterministic global dynamic optimization is quite limiting with respect to the problem size that it can handle, and this is why for the scope of this presentation we developed a surrogate model from our mechanistic one, which is a simplified model. And then we show here how the CPU times grow exponentially as we increase the number of control parameters. So the discretization of our control grid increases, and this increases also the CPU time. And we are quite limiting in the number of control parameters we can use here with this approach. But however, by doing so, for these examples, we are sure that for each of the optimizations we solved, the results cannot get better. So there is high potential in this kind of approaches, but computational powers and theory and algorithms in this still need to be further improved. And with this, I would like to move to the third question for today, which is what happens when process intensification is not all we need. For that, we took a mechanistic model for monoclonal antibody production that can also describe the glycosylation process. And we found acceptable glycosylation ranges for a typical monoclonal antibody products into the literature. And we performed a parameter estimation at the beginning to our model to have a batch case which has protein glycosylation distribution very close to our desired or acceptable ranges. And then we performed the optimization with an objective to maximize the product titer as we used previously. And we saw that by doing so, we no longer satisfy 3 of our 4 quality attributes, which is quite interesting if we consider that's what we have been doing so far and what most people do when they optimize a process without incorporating quality attributes. So then in the next step, we incorporated this acceptable glycosylation ranges into the problem formulation of our optimization problem, and we performed again the optimization to maximize the final product titer. And by doing so, we see that without having to sacrifice anything in process performance, we now are able to satisfy our desired quality metrics. Which brings me to my conclusion and outlook slide. I've talked to you about how modeling for describing process behavior can be different from modeling for optimization, and how we can use model reformulation and reduction techniques to obtain numerically proper models for optimization. I've shown to you how nonlinear and non-convex optimization problems can get stuck to suboptimal local solutions, and how we can use heuristic and stochastic approaches to obtain superior local solutions. However, only deterministic global dynamic optimization can guarantee convergence to the truly optimal global solution. And finally, I've shown to you that by optimizing process performance, we cannot guarantee that we also meet quality specifications. And I've shown you an example of how incorporating quality attributes as constraints to the optimization problem can make a step towards quality by design in biopharmaceutics. for the future, more research emphasis towards developing of hybrid models should be placed, as these models can reduce computational complexity and enable the use of deterministic global dynamic optimization methods to converge to global solutions. And I've only talked to you about glycosylation attributes as quality metrics, but there are many more considerations to be done at that point. With this, I would like to thank you for your attention, and I'm really looking forward to answer any questions you might have. If you're interested in having a look on our theoretical and numerical implementation aspects for deterministic global dynamic optimization with the use of Hammerstein-Wiener models, please have a look at our presentation. And if you're interested in finding out more about what I presented to you today, please have a look at our publication. Thank you once again.